All right, good afternoon. We'll get started. Thank you all for braving the elements. My grandmother used to say, this is a day that's good for ducks. This is, you know, maybe you may be very, most of you are much younger, and so you know these things, but when it was only about a year and a half, 2 years ago, that I thought mitochondria were the way, they looked the way I was taught and did some electron microscopy and so forth. I thought, you know, they were nice, stable things that just sat there. And of course, that's not true. And so this is a picture of the dance of the mitochondria, which is going on in every living cell except the erythrocyte, which doesn't have any mitochondria, you know. And in the process of the dance, they get longer, they move, they fragment, they come back together again. And this is a dramatic example of the impact of dynamic imaging on static biology, and even to some extent biochemistry as static biology. Okay.
What kind of cell is this one?
What? What?
What kind of cell?
I don't remember, but it looks the same in all of them. Okay, so this is today's topic of the mitochondria as all the examples are that we present, a beautiful illustration of the bridging between basic science. In this case, it's Brooklyn on this side of the bridge, and human disease, any disease. That's New York on the other side, and this, of course, is the Brooklyn Bridge. And the whole name of the game, whether it's back in the 19th century of bridge building or in the present time, is really to link the incredible advances going on in modern biology with a better understanding of conceptual physiology and its relationship to disease. And so that's the point. Now, normally we present a live patient, which we have not been doing too much this year, but will again. And the reason we do it is because you folks keep telling us that it puts a human face on a disease. So I assure you that there's every intention to continue this. It's just that some of the topics, like Ebola and dengue, and tuberculosis, they didn't quite fit with having a live ill patient. So we'll move on. Okay. So I think it's interesting, just very briefly, to reflect on the transitions in science as technologies and how they, in turn, influenced thinking about the biology and vice versa. So the mitochondria are a good example. Something was seen back in 1890s, and it wasn't quite clear what these structures were. They were done with primitive staining techniques, but they're sitting in cells. They were a mystery. In the 20th century, of course, those structures became associated with biochemistry, the classic work of Varberg, first relating to respiration, and then the discovery of the cytochromes by HeLa, and then Franz Lipmann's Nobel work on how energy is coupled to the potential. But all of this was done on homogenates and fractions, and you couldn't quite pin it down to organelles until the period of where Albert Claude, Palade, Deduve, and so forth, Novikov, created the whole field of subcellular fractionation, which increasingly permitted more purified fractions. So you really could have something that was never pure mitochondria, but at least highly enriched. And that then formed the basis of linking what had been shown biochemically with something structurally And it was around this time, of course, that the first electron microscopy emerged, which then gave a much clearer picture of what the organelle actually looks like. And in the late 1950s, Philip Sikovits coined this term, which appears in high school biology books, that the mitochondrion is the powerhouse of the cell. And then we enter a whole new area where it's discovered that mitochondria have ribosomes, genes, the genome has been sequenced, the inheritance through the female established, and these genomic structures have been done remarkable things to trace the origin, which we won't, at least I'm not going to discuss or mention anything about today, but where did mitochondria come from? Were they really bacteria that ultimately their genome was incorporated? And then we enter the area 3 and 4, which are the subject of today's exciting adventures. And that's the application of dynamic technology to discover things of how the mitochondria divide. It's by binary fission. How they move, how they fuse, how they fragment, and what does that mean? How do they turn over? How do they die? And what's the role of all of this in inheritable and acquired diseases? A huge number in both of those categories are now subjects of study, and many inheritable mitochondrial diseases have been established. Now, all of this moves in the direction of the concept, the paradigm, that if you know how it happens in detail, maybe you can figure out some way to control it, prevent it, bypass it, and that's going to be item 6 for the next 20, I suspect. Okay, so we're very fortunate at NIH to have many individuals who work on mitochondrial biology, and we're very fortunate to have 2 outstanding such ones to speak to us today. The first speaker is going to be Richard Youle, who graduated from Albion College in Michigan and then got a PhD at the University of South Carolina. Richard came here in the section of neurochemistry and has been here since 1977. He's currently chief of the biochemistry section of the surgical neurology branch of NINDS. And we put on the website, and I'm sure most of you have seen, his articles, and particularly 2 extraordinary review articles that put it in a bigger concept, particularly for people like liver doctors, or maybe like some of you who don't normally eat and breathe mitochondria. And our second speaker is Michael Sack, who took his MD and PhD in South Africa at the University of Witwatersrand. He came here to Washington University in St. Louis to be trained in cardiology, and then went back to South Africa and got a PhD in molecular biology. So he is the new species of cardiologist, the molecular cardiologist. But this is not trivial because, as you will learn, his studies in relationship to myocardial function, failure, and disease link very powerfully the role of mitochondria, and in detail, where are we in terms of understanding these processes? So, Richard, would you please begin?
Thank you, Wynn, for putting me and Michael together, my good colleague. This should be fun. So you took like my first 3 slides. Anyway, so in a cell, there's a nucleus with the nuclear DNA and numerous membrane-bound compartments and organelles, one of which is the mitochondria. It's got an outer mitochondrial membrane and an inner membrane. And what's unique about mitochondria relative to the other cytosolic organelles is it's got its own DNA. It's this circular DNA that Wynn mentioned. And it encodes 13 proteins, tRNAs and rRNAs, but 13 proteins that get incorporated into the oxidative phosphorylation machinery This is the outer membrane, and this is the inner membrane. But there's many different subunits of these components, most of which are encoded in the nuclear DNA. So this is a very interesting and complex evolutionary, coevolutionary relationship between the mitochondrial DNA and the nuclear DNA, and also a translational and a expression relationship between the nuclear genes and the mitochondrial genes. And also, since we're talking about medicine, I note that the sperm don't transmit the mitochondria to the fetus or to the embryo. These all come from the mother, and there's an interesting process during oogenesis called purification, because like nuclear DNA, these mitochondrial DNA can accumulate mutations, and that's one theory of aging, that our cells, our somatic tissues are accumulating mutations. So how do you keep this rigorous over 10,000 years? So during oogenesis, there's two processes that will select for the best or perfect mitochondrial DNA. So each one of us hopefully started out relatively pure and clean. Now there is slippage in this system, and those children have devastating maternally inherited mitochondrial DNA mutation diseases, and I won't go into that today. But there can be errors in the mitochondrial DNA itself that will lead to mutations in some of these components. Now, since there's thousands of proteins, and the mitochondria is involved in calcium regulation, a lot of metabolic processes, the TCA cycle, iron-sulfur cluster formation, so they're metabolically important in many ways. In this oxidative phosphorylation, generates a proton gradient that goes through the complex 5, generate ATP, that's why it's the powerhouse of the cell. And so how do these thousands of nuclear encoded proteins get into this, past this double membrane bilayer? This, brilliant work over decades from Walter Neupert and more recently Klaus Spanner's group and many others. In fact, I had 3 postdocs in my lab from the different leading groups around the world at one point. It's called the TOM complex in the outer membrane and the TIM complex in the inner membrane. And proteins get imported through this membrane, through that membrane to the matrix. Some of them that are membrane-spanning proteins can be embedded, stop along the way, and there can be small proteins in the intermembrane space. I'll come back to this later. And as Wynn was pointing out, this is the depiction you see in a lot of textbooks, and that's based on the EM images, and these are the cristae. This is the inner membrane where the oxidative phosphorylation occurs. Here's the outer membrane where the TOM complex allows import of proteins. And that, upon the era of EM, as Wynn was talking about, that was the kind of mentality. And with the recent, the big advances in confocal microscopy or light imaging, it's more apparent to all of us that they're not necessarily bean-shaped, that they have this heterogeneous morphology like Wynn showed in his movie, and I'll just show another movie of 2 different cells, and these are HeLa cells. And you can see they're quite mobile and they move around, but what you'll note is they have different morphologies. Some are little round things, some can be very long and tubular, and this, as Wynn says, kind of an interesting new topic. People think it's kind of curious because we used to think they look like this, and now we think like this. But in fact, before EM, when these were discovered 100 years ago, they used light microscopy, and they knew this already. And in fact, that's embodied in the name of mitochondria, because this is Greek for mito, means thread in Greek, and chondria means brain. So 100 years ago, they knew they had this plastic heterogeneous morphology. But what they didn't know 100 years ago, and what's fueled a lot of the interest, is some of the genes that are regulating this morphogenesis have been identified primarily in yeast by Janet Shaw and Jody Ninari, and in C. elegans by Alex Vanderbleek. These were some of the pioneers that discovered these 3 types of large GTPases. PRP1 is involved in fission. It is involved in fission of both the inner mitochondrial membrane. So you have this inner membrane gets divided, and then the outer membrane gets divided. And in fusion, you have this mitofusins that are involved in fusing of just the outer membrane. And you've got OPA1 it's in here that's involved in fusion of the inner membrane. And why I'm bringing that up, because of the focus on the medical links to mitochondria, is that mutations, so initially you think, well, why do they bother to do this? What is all this fission and fusion? And let me just back up a minute. I forgot to show the movie. So this is that same movie I showed you where they're wiggling around, but just notice these couple little spots right down here. See this mitochondria tubule sort of, it's going to break off in a second. See how it broke off there? That's a fission event. Then it broke in half again. Now this one just broke off, and watch, these two are going to fuse. So they're going to undergo this fission and fusion process. The question is why? Is this important for anything? And if so, what? And so the reason we know it's important is there's two fusion genes, mitofusin 2 and OPA1, have been identified to have genetic links to neurodegenerative diseases. Mitofusin 2 causes a peripheral neuropathy, CMT2A, and interesting, a mutation in OPA1, and actually it's simply a haploinsufficiency, just loss of one allele of OPA1 causes the most common hereditary form of blindness called dominant optic atrophy. So there's two genes in the fusion pathway just showing this is an essential process, and what I'm going to talk about is why. And it's interesting and curious why this different cell type selectivity of two fusion genes, as in many mitochondrial diseases, is not known, but it does show the importance of this process. So it's a little bit hard to study if you just look at cells and you can count events. It's quite tedious. So I had a postdoc, Mariusz Karbowski, who developed a very nice quantitative method to assess this. And he did it shortly after this groundbreaking work here at the NIH by George Patterson and Jennifer Lippincott-Schwartz, who developed the photoactivatable form of GFP. So Mariusz got it from them like within days of that's one of the nice things about being at the NIH. You see a paper in Science and you go over to their lab the next day and you get a construct. And he fused the photoactivatable GFP to a mitochondrial import sequence so he could target it through the TOM and the TIM pathway to the matrix compartment. And this is a HeLa cell that has been transfected with a matrix-targeted photoactivatable GFP. So you can see the mitochondria throughout the whole cell, but as George and Jennifer showed, if you shine a laser on this, you can photoactivate it, and you can make a very small laser, just maybe twice the size of that pointer, in these two spots, and it instantly, within milliseconds, fills the volume of that tubule. It'd be hard to say in here where the beginning of one mitochondria is and where the end of another, but as soon as you see that filling, you can easily tell where that one ends and where the next one starts. But then you can watch this over the next 30 minutes, and you can quantify the fusion rate by looking at you look at the equilibration of this photoactivated GFP throughout the whole network. So now this is over 30 minutes. So you see the fusion events, and you see it just dilutes it out. And here down here, you'll see the next one. And then generally in HeLa cells, within 30 minutes, this is equilibrated throughout the entire cell. And remember I mentioned this OPA1, like mutations in this cause blindness, and it's involved in fusion. So if you do RNAi to knock down OPA1, let's do the same experiment. Well, first you see this cell with an OPA1 RNAi. The mitochondria are all much more punctiform. But then watch the fusion process comparing those two over the same time course. So these completely equilibrate. First of all, these don't move around very much. They're little fragmented dots because they can't fuse, and the photoactivatable GFP does not dilute out. These are some new techniques to quantify these processes. Now let me just think. So, okay, so I covered fusion. Now DRP is another, it's in the same family, but it's involved in the fission process. And really important work was done here at the NIH and initially by Alex Vanderbeek's group at UCLA, where he was able to GFP label DRP and watch it in real time. You see it's actually little green dots on the red mitochondria. And here's some time-lapse images where he goes from this moment to this moment. And look right here. You got this little green dot assembles on the red mitochondria. It's bigger. There's a breakage, and you have a division event. So in other work by a number of groups shows it forms a spiral around the mitochondria and causes scission. And if you knock down DRP or knock it out, the mitochondria in green become highly interconnected. This is the opposite of that OPA1 mutation. They all come together. And one, just kind of to update, the most recent advances come from Gia Volz's lab at the University of Colorado in collaboration with Jody Nanari at UC Davis. And they've found this incredible process where ER tubules will wrap around mitochondria and demark points of subsequent fission. And it's not known the molecular biology or even the reason that this happens, but it does clearly happen. And it may stimulate the formation of this DRP1 spirals in fission. And so that's like the latest important topic in the field for what does that do, how does that trigger fission, and how did that ever come about? So anyway, so what I want to focus now on, why do mitochondria fuse? So this is not so obvious. They clearly have to fuse to maintain health. And one theory is if there's some damage or if there's some missing, if this is deficient in one substance or protein and this is deficient in another, if they fuse, they may complement one another's activity by equilibrating substances throughout them, because it's clear that fusion is stimulated by two processes. Increased energy demand causes energy demand, and also by stressful conditions cause fusion. Now, fission's easier to understand in some ways. I mean, obviously it's understand you need to divide mitochondria to repopulate more cells. Every time you have more people on the planet, you need more mitochondria, so that you have to divide. But also, what I'll come back to later is this idea of fission as a quality control process. And this was first noted by Orion Shirahai at BU. And he noted that after certain fission events, one of the daughter cells would become degraded by autophagocytes, leading to his ideas and some of my extension of those ideas that you've got a mitochondria, and as it undergoes biogenesis, particularly if you exercise, eventually there may be some misfolded proteins and damage accumulation with time and activity of the mitochondria, or reactive oxygen species generated by oxidative phosphorylation. And as long as this debris will aggregate, a fission event anywhere will give you some kind of segregation of good and bad. And there's examples of this in what do you call it, in rod-shaped bacteria, in Bacillus. It's known that two daughter cells of bacterial division are not equal, that one will be more rigorously maintained for future generations, and debris will be accumulated in one of the daughter cells. So the same might apply here. But I'm going to come back to that because recently some of the genetic mechanisms of this has been worked out and linked to disease. But first I want to go to one of the other interesting processes that occurs during fission, and it occurs during apoptosis or programmed cell death. So I think of this programmed cell death process as a reversible phase. We get a lot of signals, a commitment step, and then the irreversible phase. And a lot of things happen in the irreversible phase, like activation of proteases, activation of nucleases, disintegration of the whole cell in a very programmed, non-inflammatory way. But the decision point is when one of these BCL2 family members called BAX, it translocates from the cytosol of the mitochondria, causes release of cytochrome C from the mitochondria to the cytosol, and that activates caspases. And interesting, at the same time, mitochondria divide excessively. Now, I mentioned there's these BCL2 family proteins that regulate cell death. These are anti-apoptotic. These are pro-apoptotic. And I had a postdoc and a medical student who made this really interesting finding maybe 10 years ago that relates to this commitment step. Because before BAX translocates to the mitochondria, you can reverse this process, but afterwards you can't. You can prevent these processes, but the cells are doomed in any case. So what this medical student, Keith Walter, did is he fused green fluorescent protein to BAX, and he labeled these 5 cells. And 2 of them have been transfected with the RFP fused to a mitochondrial import sequence. So there's 2 cells that have got the mitochondria labeled. There's BAX in the cytosol of all of them. And this is just what BAX looks like in healthy cells. You see it's just in the cytosol, very diffuse. But if you induce apoptosis, watch what happens over 30 minutes. Now look at that change in the BAX distribution. You can't really tell where it is, but if you look at it, where the mitochondrial label, it translocates to the mitochondria, forms these very punctate spots on mitochondria, And that is when cytochrome C is released from the mitochondria. And what's interesting is if you look at those spots in detail, so here's a blow-up of one mitochondria in green and the backs is in red. And this is done by Mariusz Karbowski, an expert microscopist. And you can see over seconds that there's little red dots of backs form on mitochondria, and actually they assemble at constriction sites. Or over here you can see a number of these little red dots over 3, 6, 10 seconds. You see those two come together and the mitochondria breaks off. Remember those old division sites? And there's backs on the tips. Here's some more static images and some E images showing that this backs, this backs going to mitochondria assembles at the tips or constriction sites of the mitochondria. You can also see this by immuno-EM. And remember this images of DRP I showed you, where DRP is going to focal spots of mitochondria that assemble at constriction sites and become division sites? Well, these co-localize with BAX. So BAX is going to these DRP sites. Now, as I mentioned, cytochrome C is released. That's one of the BAX translocation cytochrome C releases the points of no return. So here's mitochondria. This is a field of cells that have been induced to undergo apoptosis. So you're looking at just kind of a whole population of cells. Cytochrome C is stained in green. So these are mitochondria where the cytochrome C is still in the mitochondria. Here's a cell where it's completely released. You can't even see where the mitochondria is. But look at these three cells. They're partway through the whole system. They clearly have cytochrome C still in the mitochondria. Some of it's released here, but you can see that the mitochondria here are fragmented. That's what I'm talking about. Corresponding to Bach's translocation of the mitochondria at constriction sites, fission occurs, mitochondrial fragments, and that you never will find a tubular mitochondria with cytochrome C release. So it's linked to this process. And if you inhibit DRP, remember I showed you that very interconnected cell where you inhibit this division protein? You prevent that fragmentation, but then look at the mitochondria are in red, and look at when the Bach's translocates in these green spots, and you can see so many of them are on tips and constriction sites. And here's an immuno-EM of Bach's on one of these mitochondria that is prevented from dividing, but you see the Bach's assembling in these large spots. And still, this is like a mystery point. I've struggled for this for years now. What do these represent? What's the nature of this? And this is probably where cytochrome C is coming out. And then I just want to, how long should I go? Till like 4:45? So now I want to come back to this, because this is the current main focus of my lab. And it's serendipity, but 2 genes mutated in familial Parkinson's disease have been linked to this process of mitochondrial autophagy. You smart guy. So there's only 3 known monogenic autosomal recessive causes of Parkinson's disease. There's some autosomal dominant causes like alpha-synuclein or like LRRK2. That's like Huntington's disease is a dominant. Those are probably, you get 1 copy of a mutant gene and you get the disease. And a lot of those are thought to be gain-of-function mutations, like a misfolded protein. It's doing something bad because you've got a good copy and a bad copy, and if you get the bad copy, you're going to get the disease. These are autosomal recessive. And I'll tell you, in most of those cases, I can't think of one case where an autosomal dominant, people have figured out the cause of disease. We don't know what causes Huntington's or alpha-synucleinopathies. These are maybe more tractable because they're autosomal recessive. They're very likely loss-of-function mutations. So you don't have Parkin, you get Parkinson's disease. You don't have Pinkone, you get Parkinson's disease. So at least you can think about what do they normally do. You don't have to imagine what the misfolded protein might do. And here we've got a lot of hints because Parkin is an E3 ubiquitin ligase with ring domains. And it's known to be in the cytosol. This is before we worked on it. And Pinkone was known to be a kinase, or at least it had a clear kinase domain. And it had a mitochondrial targeting sequence. And that led to Nick Wood and the people who discovered it's mutated in PD to look at it in cells, and indeed it's mitochondrial. So then there's a third one called DJ1. And I don't know, where is DJ1 in the cytosol? David, where's DJ1 in the cytosol? So there's another one, DJ1, that's an autosomal recessive. So the question is, what do these do, and is there any relationship between them? and the field percolated along for a while, and because then people made knockout mice, no phenotype. And people with mutation, autosomal recessive mutations, neither. Those get phenocopy one another with Parkinson's disease. The mice knock out Pinkone, both alleles, knock out Parkin, no phenotype. But at least they phenocopy one another. But then Leo Palank moved the whole field forward in my mind when he looked at Drosophila, because they had a phenotype. And like a mitochondrial disease, you can imagine mitochondria in different species are important in different tissues. You can imagine, you know, like bumblebees aren't supposed to even be able to fly. They probably have flight muscles, are very aerobic, and eagles probably have really a lot of energy demand on their eyes. But anyway, so the flies had a flight muscle defect. So then when he made these Parkin mutant flies, and then he did EM on those flies, and the mitochondria were swollen. So that was interesting, because here's a link of this cytosolic protein to mitochondria, and this one's already been localized to mitochondria. Then a couple other labs stemmed from that. They did the same thing with Pinkone, and it phenocopied the Parkin in the fly with a flight muscle defect. And then they did epistasis, this genetic experiment where you put them together, and it's not worth suggesting they're in the same pathway. So the question is, what is the pathway? Do they function together, and what do they do? And I'm just going to now summarize a bunch of work from our lab and other labs over the last 5 years. But it was stimulated by a graduate student, Derek Narendra, in my lab. So we and others have found one thing that Pinkone and Parkin do together, like a molecular cell biology pathway, where they function together. We knew they genetically function together, but this is one thing they do together. And I'll come back at the end of how this may be related to Parkinson's disease. So if you've got a cell, and it's got a couple mitochondria, and one of those mitochondrion gets damaged, what happens is this kinase, Pink1, accumulates just on the damaged one. Now this is quite, how can you imagine that would happen? How does Pink1 know? It's got a mitochondrial import sequence. It should be imported into all mitochondria. And I'll show you this in a minute. And then Pink1's a kinase. Well, what it does is that through its kinase function, it recruits Parkin from the cytosol to the mitochondria and also activates Parkin's E3 ubiquitin ligase activity. Parkin's an E3 ligase, but it's in the off state. Pink1 activates it, recruits it to the mitochondrial surface, and it ubiquitinates lots of proteins on the outer mitochondrial membrane. And these chains of ubiquitin stimulate autophagosome assembly and elimination, suggesting that if the mitochondria gets damaged and then eliminate, maybe this is a quality control pathway. And this is really important because if this is true, and if it's all that they're doing, it suggests an etiology of Parkinson's disease. So I'll give you some of the evidence for it. Well, this is what my graduate student Derek did first. You can imagine he came to the lab. I'm in a neurosurgery department. He's in the middle of medical school. He wanted to do something clinically relevant. We're working all this heavy-duty mitochondrial fission fusion. But we've been watching the work of Mark Cookson and others in our building working on this pathway. And we thought we might just try our old trick. Remember I showed you Bax translocates when we fused GFP to it? So I suggested to the medical student, why don't you take Parkin and fuse GFP to that and see what happens? And he did just that. And you can't see it well, but maybe this is worth turning the lights down for. But they changed the program, so we can't do that. It's all right. It'll become very clear. So here's two cells. The mitochondria are stained in red. The GFP is fused to Parkin. And just like the literature says, it's definitely in the cytosol. So we induced apoptosis. That was the first thing we did. Bax translocates, maybe Parkin translocates. Nothing happened. Then Derek thought, well, let's add paraquat, because paraquat's an herbicide. It's been linked as an environmental insult that has increased. There's increased PD in farm workers, and people think paraquat may. So he added paraquat, and he saw some very strange things happening. But paraquat's a pretty strong oxidizing agent, so the cells look trashy, a lot of things go wrong. So he started playing around with other things. He found a very simple, non-toxic, reversible step called adding a mitochondrial uncoupler. Remember in that second slide, OXPHOS generates a proton gradient across the inner and outer, the inner mitochondrial membrane to drive ATP. An uncoupler is something that just reverses that proton gradient. How much time have I got? You got time? An uncoupler reverses that proton gradient. For example, dinitrophenol is an uncoupler. It's been used in humans to promote weight loss. There's another uncoupler we use a lot, CCCP, valinomycin's another one. And so then he added this uncoupler CCCP, and watch what happens to the green Parkin. After he tried many things and nothing would change, he now sees very robust translocation of Parkin to the mitochondria. And that's what I mentioned in that review slide. It's a very robust result. And we knocked down or knocked out Pinkone, and that completely prevents that Parkin going to the mitochondria. There's like 100% effect. So this links these two gene products mutated in PED in the same pathway. Pinkone recruits Parkin to the mitochondria. And patient mutations in Parkin and Pinkone have reduced or completely blocked translocation activity. So what I'm going to get to now is the whole pathway, and then what's the link with Parkinson's disease. Okay, so this is adding a chemical uncoupler. What does this mean for normal human biology? So we looked at some other things. We looked at some cell types. Remember I was talking about mitofusin 2, the mutations that cause Charcot-Marie tooth disease and are involved in mitochondrial fusion? Well, we knew a paper from David Chan at Caltech who'd made knockout mice. He knocked out MFN1, he knocked out MFN2, and he made double knockouts. They're all constitutive lethals. You don't get mice. But he made mouse embryonic fibroblasts from these animals, and he generously shared them with us. And the reason we wanted them is he had a paper in JBC some years ago, and he looked at the mitochondrial membrane potential. So the uncouplers uncouple the mitochondrial membrane potential. And this paper said that the MFN1, MFN2 double knockout has about 10% of the mitochondria are uncoupled, but the cells are viable. Whereas none of the mitochondria are uncoupled here or here. So we thought, well, that's interesting. Let's think of, instead of adding a chemical uncoupler, what about a genetic model where a subset of the mitochondria are damaged, not all of them? So we found this mitochondrial stress other than chemical depolarization causes Parkin translocation. Because you look at wild-type cells, let's say 98% of the cells look like this. Same here, but in the double knockout, most, by far, 90% look like this with these little spots of Parkin. Well, what do those spots represent? So here's the same type of setup, MFN1, 2, double knockout cells expressing GFP Parkin, also stained for cytochrome C to label all the mitochondria and mitotractor to measure membrane potential. So here's cytochrome C staining in this square right here. You can see all the mitochondria. And here's Parkin. You can see right off the bat that there's Parkins not on all the mitochondria. And if you look at these two, for example, right, these two mitochondria stained for cytochrome C, you look on the right side, one of them has lost membrane potential based on mitotractor staining, and one is not. And then if you look over here where the Parkin is, Parkin's on the other one. Here's another example in the same field. Here's a mitochondria, lost membrane potential, there's the Parkin. So it seems that Parkin can somehow identify the one damaged mitochondria out of 50. How does it do that? And so now a lot of papers have come out since then. A lot of different uncouplers have been shown to cause Parkin translocation. A lot of genetic models. I just showed you MFN knockout cells, paraquat I mentioned. But even unfolded proteins without any uncoupling of the mitochondrial membrane can cause Parkin translocation. So what do these have in common? What's the common denominator? The common denominator is Pinkone accumulation on the outer mitochondrial membrane. So how would that work? So, you know how some of you, like postdocs, mentors, I suggest something, and then they, of course, say, that's crazy. I'm thinking kinases are turned on and off. And since I have some guys and gals that are really good at Western blotting, a lot of people see it, let's just do a Western blot and see if it's there or not there. What are the chances that's going to work with a kinase, right? Kinases are there, they're turned on and off. So they do it anyway. And so they blot for Pinkone. And you look at HeLa cells, this is endogenous Pinkone in HeLa cells, you can't see any Pinkone. And you look at the literature, people already knew this. And they said, well, because everybody's saying, oh, the Pinkone antibodies don't work. But then they add CCCP. This is that uncoupler that causes Parkin translocation. And you see a really beautiful band right at the right exact molecular weight of Pinkone. So we talked to our colleague, Mark Cookson, downstairs. He had some nice RNAi for Pinkone because people would say, well, that's not even Pinkone. That's some kind of off-target stain. But RNAi Pinkone completely rubbed it out. This is indeed Pinkone. The reason you can't see it is because, Well, why can't you see it there, and why can you see it here? Well, I told you, CCP is easily washed out. If you wash out the CCC for 2.5 minutes or 5 minutes, it very rapidly disappears, meaning pink one's rapidly turned over. So we screened all the proteases in the mitochondria, and we found one called PARL. We knock it out, we got knockout meths, or you knock it down, it prevents this constitutive degradation. There's a little band below it, I'll explain in a second. You add CCCP, the lower band goes away, but nothing changes in the upper band. You wash the CCP out, the lower band comes back again. Anyway, PARL is a rhomboid protease. That's a class of proteases that are membrane-spanning, multi-spanners that cleave membrane anchor domains in proteins. And PARL specifically localized to the inner mitochondrial membrane. So this, put together with a lot of data I don't have time to go into, leads to this model. That there's a mitochondrial targeting sequence. Remember I showed you in that first diagram of pink one, that's how people thought it was a mitochondrial protein, because it's got a mitochondrial targeting sequence that targets it for the TOM. Remember in the very beginning I showed you the TOM complex and the TIM import pathway? So this is normally cleaved on imported proteins by the mitochondrial processing peptidase, MPP. That's what this is. When it gets imported, it gets clipped by MPP. Then, unlike any other protein known in the mammalian membrane anyway, PARL has substrates known in yeast, but not in mammals. This is the first one. It cleaves right about there in the membrane spanning domain of pink one. And then I had a very clever Japanese postdoc, Koji Yamano. And we knew people had shown this earlier. You know, you couldn't see pink one, but if you add MG132, a proteasome inhibitor, then you could see a band of pink one, but it was the wrong molecular weight. Instead of being 62 kD, it was 52 kD, which exactly matches this. And so then a group in London sequenced this, and they reported the sequence as a phenylalanine. And then my Koji Yamano goes, and we still couldn't put it together. Oh, it cleaves there, generates a phenylalanine, and it's degraded by the proteasome. And Koji goes, well, that's the N-end rule of Warshawski, because phenylalanine is an N-degrand, susceptibilize you to these three E3 ligase and proteasomal degradations. And he indeed perfectly showed you mutate that, it's stable, et cetera, et cetera. So it's very clear. Pink one is imported, clip, degraded by the proteasome. And that sum total generates this, basically very low steady state of pink one. Now, you may not all know, but if you uncouple mitochondria, you block import through the TIM pathway, but not the TOM pathway. This is prevented by uncoupling. That's well known by Neupert and all the mitochondrial import people. So if you block import of pink one, it's never accessible to PARL. And it's shown a very good a biochemist named Michael Lozaro, who's back faculty in Australia. He showed that this is 100% bound to the complex. And Sam Hassan showed it was required 100% requirement of one of the subunits of the TOM complex called TOM7. So this is bound to the TOM complex. Why that is, I've got a postdoc trying to figure that out right now. But anyway, it does. And that is this right here. So this is bound to the TOM complex when you have CCC3 present to prevent the import here. And from there, the kinase function recruits Parkins to the mitochondria. So that explains, it's really interesting how nature sort of figured this out. You know, how could pink one accumulate just on the damaged mitochondria? It's because the healthy mitochondria are constitutively importing it and degrading it, and import is some kind of sensor for damage, and that allows it to accumulate on the outer membrane and recruit Parkin. So what's pink one substrate? How does pink one recruit Parkin? This is the most exciting thing, and we published a paper last year, and along with a lot of other people. So at least it's reproducible. I won't go into the whole story. I'll just cut to the chase. Pink one had been shown by this group. This is called the group of Muckett in Dundee. I mean, a lot of people say, "Was pink one phosphorylate?" Well, the first semester answer would be maybe it phosphorylates Parkin, and there was paper after paper said it did. And we couldn't repeat any of those data. But this paper in 2012 said, oh, pink one phosphorylates Parkin. And this paper is the first one we could reproduce. It was serine 65. But we mutated that or deleted the whole domain. Is out here in the very C-terminus. Is this ubiquitin-like domain. You know, ubiquitin is this post-translational thing that's stuck on proteins, made chains. some proteins have something that's homologous to ubiquitin in different motifs. And Parkin indeed has one of these. So these guys said, oh, it's ubiquitinated serine 65. So we mutated serine 65. We deleted this whole domain. Pink1 still recruited Parkin to the mitochondria, still with its kinase domain. So we knew there had to be another substrate. So Leslie Kane, first author on this paper, just about a year ago, she did a really nice mass spec study. It wasn't easy. She'd been trying for 4 years to find the substrate, basically comparing mitochondria, because we knew it should probably be on the outer mitochondrial membrane. So she compared plus and minus CCCP. Never could figure anything out. Then, in the last year, CRISPR technology came on board. We're really good at CRISPRs. So we knocked out Pink1 in these HeLa cells. It's the endogenous Pink1. We knocked out Pink1 and added CCCP to both. That and some other clever tricks Leslie used on mass spec. She identified a substrate, and as soon as we saw it, we knew it was the right thing, because she found it was ubiquitin itself. That Pink1 phosphorylates ubiquitin, and it's serine 65, the same serine that these guys, and we'd confirmed, Pink1 phosphorylates in the ubiquitin-like domain. And so we could obviously make phosphoubiquitin in a test tube. You can just buy ubiquitin, phosphorylate with Pink1. You mix that with Parkin, and it activates its E3 ligase activity. So we had the paper in April. These people had the same idea, and they published another paper within a month of ours. And then the Matsuda group in Japan had a paper the same month. And then there's 2 other really big studies have come out all in the same year. This is from the Wade Harper group, and that's from the Commander group, all showing these both are possible, but clearly phosphoubiquitin is sufficient. So that leads to a really interesting model about how this whole thing works. Because we've been working on this for many years. There are ubiquitin chains on outer mitochondrial membrane proteins. There's a process of turnover of outer mitochondrial membrane proteins going about its own business. But then when Pink1 accumulates after damage, it can phosphorylate these chains. And then these chains become receptors for Parkin, and Parkin binds to these chains, and it activates. And Parkin's an E3 ubiquitin ligase. So Parkin puts chains on the mitochondrial outer membrane. So that leads to a very strong feedback amplification. You just reiterate that. So there's some ubiquitin, already amide-linked outer membrane proteins. If you have damage, Pink1 becomes stabilized and phosphorylates that ubiquitin. This then binds and activates Parkin, and Parkin ubiquitinates the mitochondria that stimulates mitophagy. But it's putting ubiquitin on the mitochondria that in turn is a substrate for Pink1 to generate more phosphoubiquitin, and that's likely what drives mitophagy to completion. You don't go partway down this pathway. Once you get this thing started, the feedback amplification. And then the last thing, if I've got time, is how do these ubiquitin chains signal autophagy? And I really haven't talked about autophagy. I just said it, and I showed some pictures of it. So what's the real evidence that there's autophagy of the mitochondria? Well, the one piece of evidence comes from Japan. After we had found this pathway, Mizushima's group. Mizushima's a leader in the autophagy. He looked at Parkin-mediated mitophagy. He's very good at EM of autophagosomes. And he's got a nice paper with a series of images like this where it's not 100% selective, but there's relative selectivity for engulfment of these clustered mitochondria following Parkin activation. So we thought, I don't know how much you know about it, but there's a series of adapters like P62, proteins that are known to bind ubiquitin, like via UBA domains or UBAN domains, that also have a domain, a LIR domain, LIR, that binds to LC3. LC3 is a component of the autophagosomal membrane. And P62 is thought to be a link between ubiquitinated proteins and autophagosome. So we knocked out P62, and we didn't see any phenotype. Then we knocked out another one, didn't see a phenotype. So we decided to knock out at the time, all 5 known adapters. And this is what we found. And I'm just going to back up for a second. So how do you measure mitophagy? Here's one way. So here's a group of HeLa cells stained for mitochondrial DNA in green and counterstained in blue for the nuclear DNA. See all these little green speckles in the cells? That's the mitochondrial DNA. And if you add an uncoupler for 3 hours, you see the mitochondrial DNA gets a little clumpy, but by 24 hours, the green staining is almost gone. So is that autophagy? So we knocked out ATG5. This is a canonical autophagy gene. And in the ATG5 knockout HeLa cells, you see the green dots. You can see they clump up quite a bit at 3 hours. They're even more clumped at, they're even more clumped at 24 hours, but they don't disappear. So then we knocked out all 5 of these adapters, and we looked at this mitophagy pathways here. So here's we call it the 5-tail, and you see this the green speckles, and here it's not as clumped here. It's definitely not disappearing. So you need some of these adapters, and I'm not going to go into a lot of details, but optineurin and NDP52 seem to be the most important two. And then I'm just going to wrap up here and just kind of summarize what's kind of interesting about this step is there's a new mechanism of kinase regulation, the import conditional degradation, and there's a new mechanism revealed of a new way to activate E3 ligase using phosphoubiquitin. So it's very similar to a substrate activation of an enzyme. Some of the big questions are, does the absence of this pathway, is this causing Parkinson's disease? That's quite important because of the clinical ramifications. Why would such a pathway be selective for certain types of neurons? No clue. And then a lot of interest now in, can we stimulate this pathway to treat mitochondrial diseases or Parkinson-related diseases? And then I just got to summarize the whole talk just as a reminder. Mitochondrial morphogenesis and dynamics, mitochondrial fission fusion linked to disparate neurodegenerative disease, CMT2A and DOA, role of mitochondrial in apoptosis, and mitochondrial quality control linked to genetically to PD. And then I tried to mention all my postdocs along the line, so I'll just show a picture of them and mention this is my group now, one graduate student right there, and these are the former postdocs I've had that have contributed to the slides I showed. Thank you.
We have time for a few questions, and then we'll have more at the end.
Hi, nice talk. I got a quick question. When you depolarize mitochondria, long-term depolarization, how do you do that? Because actually you cannot keep these guys depolarized for long. They're going to go for mitophagy, the whole organelle, I mean. And the thing is that even if you try to create raw zero cells with, ethidium bromide, it's not like mitochondrial DNA completely vanishes, it's diminished. So do you think that there is some kind of selective mechanism to get rid of the bad mitochondrial DNA and the cells keep some pool of the healthy DNA? It's just a speculation and you don't have anything in mind.
Yeah, I mean, it clearly, if you've got a very deleterious mitochondrial DNA mutation, we collaborated with I'm blanking on the guy's name, but complex 1 mutations, Parkin will selectively shift the heteroplasmy from mutant to wild type. Sure. But for other ones, we have not seen any purification. So we just did a big study and submitted now, where we looked at the mutator mouse. The mutator mouse has got a mutation in polymerase gamma, the mitochondrial DNA polymerase, and it's a mutation in the proofreading domain. So these mice accumulate a big variety of mitochondrial DNA mutations. And these mice don't live as long. Niels Gorhan Larsen made them, Tom Pola made them. They have a shortened lifespan, they look old, they're thought to be an aging model, I don't know about that, but there's a phenotype. So we took those mice where we know the cause of disease. We don't really, anyway, we know there's a mitochondrial disease. And we crossed them to the Parkin knockout to look at the role of endogenous Parkin in the mutator mouse. And we saw a synthetic phenotype, meaning they did worse in the absence of Parkin. And we could rescue that with L-DOPA. So we did some really heavy, deep sequencing of the mitochondrial DNA. So we have good evidence that Parkin, endogenous Parkin, rescues those mice to a certain extent. But we did the deep sequencing of the mitochondrial DNA. It does not rescue sequence. So I think, I just kind of go with the flow. I mean, I would have thought what you thought, and that's why we did the experiment. So maybe it's that Parkin is rescuing downstream of mitochondrial DNA damage at the protein level, at the protein aggregation level, to compensate for damaged proteins or mutant proteins and rescuing them perhaps by some kind of selective segregation of mitochondrial debris and elimination by mitophagy. And if you don't have that process, the mutations lead to more gumming up more rapidly.
I'm just wondering how the cell can sense where the bad mitochondrial DNA is.
Based on Pinkone. Yeah, they can't, you're right, they can't really tell where the bad DNA is because of fusion, unless it's a super deleterious, one of the big complexes that don't diffuse well that kind of stay near. tRNA mutations, they never find them, right? It's like in 30 minutes, it's everywhere. So it doesn't purify, by and large, at the DNA level. Downstream, probably.
Richard, that was a tour de force and a really wonderful overview of the molecular characterization of mitochondrial function dynamics and obviously looking at genetic diseases. I'm going to take a different tack now and go into acquired changes in mitochondria and you said essentially with respect to nutrient overload or feeding and fasting and the effect on human diseases. Okay, so I'm going to try and keep these buttons correct. But so essentially, today I'm going to talk about caloric load, a mitochondrially enriched deacetylase that deacetylates lysine residues of mitochondrial protein, SIRT3, and the regulation of protein acetylation. And look at that in respect to a specific disease, and that's the role of fasting and liver toxicity due to Tylenol. So I'll go back to one specialty here of liver disease. And then look at certain biology more globally and its role in mitochondrial quality control. And then talk about a new study in the lab, which is unpublished, looking at the role of fasting and SIRT3 on a mitochondrial quality control immune program called the NLRP3 inflammasome. So just a bit of background here. I think we all know that nutrient excess and obesity are creating increasing disease burdens. And if we just look at the US from 1980 to 2008, there's really been a huge increase in this process. And these data are from Minnesota, but essentially showing or suggesting that 50% of excess deaths due to cancer, heart disease, stroke, diabetes, etc., is due to obesity, and there's a huge financial burden on every single state based on that. So this is just an example of how nutrient overload can affect intracellular programs and pathways, and this is looking at growth. And so either excess glucose, amino acids, or not shown in the slide, fatty acids, can increase intracellular substrates. I'm not going to talk about the glycosylation pathway, but increased glucose can cause glycosylation of growth factors to drive this pathway. But either glucose or fatty acids can generate a metabolic intermediate acetyl-CoA, which in this schematic, through the citrate shuttle, gets out of the mitochondria and can be used, the acyl group, to acetylate histones to then activate genes involved in growth, and amino acids through mTOR, PI3 kinase A and T. So together, these metabolites can drive a lot of intracellular processes in the cell. So the histone acetylation brings up the topic of lysine acetylation, and just to give you a brief history of that, the first histones were discovered to be acetylated in 1968, and in 1997, the first non-histone protein or acetylation of a protein was found, and that was P53. Before 2006, so really just in the last decade, there were only about 90 proteins known to be acetylated. And then this paper came out by Kim et al. In Molecular Cell, and this, I guess, this advance is sort of aligned with increased mass spectroscopy in terms of looking at these post-translational modifications, where they found 196 proteins, and now there's in excess of 2,000 acetylated proteins. And the most interesting thing about this study in 2006, was that they noted that there was a difference in which proteins and which lysine residues were acetylated depending on the fed or fasted state, and this is in the liver. And they also noted there was a high enrichment of acetylated proteins in the mitochondria. And this is an immunoblot from the liver from the Kim paper in 2006, and they show that some proteins which were acetylated in the fed state, this is an antibody to acetylated lysine, are deacetylated in the fasted state. And this is a study from our lab showing that if you look at a chow diet versus a high-fat diet, also in the liver, there's increased mitochondrial protein acetylation in response to nutrient overload or high fat. And so with respect to this regulation of acetylation and deacetylation, there could be some non-enzymatic processing, but there's also an active enzyme system, and acetyl transferases acetylate substrate proteins on lysine residues, and deacetylases are NAD dependent when they deacetylate proteins to produce a deacetylated substrate plus nicotinamide and the acetyl group. And nicotinamide actually, interestingly, inhibits this deacetylase enzyme. So there's 7 sirtuin deacetylase enzymes, and these are distinct from the histone deacetylases, which are zinc dependent, and these are, as I alluded to earlier, NAD dependent. In mammalia, there's a SIRT 1 through 7. They showed distinct subcellular localizations, with 1 and 2 in the nucleus and cytosol, 3 to 5 in the mitochondria, and 6 and 7 again in the nuclear. And this just shows the acetyl group on the epsilon amine of the lysine residue. And with deacetylation, there's a charge change from positive to neutral. And this lysine residue could also then be amenable to other post-translational modifications such as ubiquitination, stimulation, methylation, etc. And interesting, this acyl group creates a bromodomain which facilitates protein-protein interactions. So SIRT3 is found to be the nutrient-sensing mitochondrial deacetylase, and here we see a knockdown of SIRT3, SIRT4, SIRT5, the 3 mitochondrial family members, and you only see increased acetylation in the SIRT3 knockout. And this is from David Lombard's work when he was in Fred Alt's lab in Boston. And in fact, to show that it's nutrient sensing, this is data from our lab showing that on a high-fat diet, you get decreased SIRT3 levels. And this is from Eric Verdon's group demonstrating that fasting, quite acutely, up at about 12 hours to 48, there's increased SIRT3 levels in the liver in response to this change in nutrient status. So this is coming to the story of acetamin, which is Tylenol, or and hepatotoxicity. And this is a study from 1994 out of the University of Pittsburgh, and they followed patients that developed acute acetaminophen-induced liver injury. And as you know, on the Tylenol box, there's a black box label to say, don't take Tylenol with drinking because alcohol can predispose to liver failure. But what they noted here, that at a much lower dose, 4 to 10 grams of acetaminophen a day, you're much more likely to develop liver injury if you're in the fasted state versus the fed state. And only above 10 grams there was a greater susceptibility to ethanol use. And they really didn't understand why this was. And interestingly, it's not on the black box label that you shouldn't take Tylenol when you're fasting. Nevertheless, it's also interesting to note that a major metabolite or toxic metabolite of acetaminophen is NAPQI, which is N-acetyl-P benzoquine imine. And NAPQI can either bind to cysteines or to lysine residues, and it's a very oxidative metabolite. It depletes the glutathione pool in mitochondria. resulting in junk activation, reactive oxygen species, mitochondrial permeability. And so it's recognized that NAPQI is a toxic metabolite, and it can potentially, and it's been shown to bind to lysine residues. So from that, we hypothesized as could the level of mitochondrial protein acetylation modulate susceptibility to acetaminophen liver injury? And could this be mediated in part by modulating NAPQI binding to mitochondrial proteins? So we first looked at acetaminophen injury. The chemical name for acetaminophen is N-acetyl aminophenol, so that's APAP. And interesting, when we compared the wild type to the SIRT3 knockout mice, in the fed state, there was really no difference in release of liver enzymes, which is a mark of liver stress. But in the fasted state, the knockout mice, the ones that have increased protein acetylation, seem to be protected. And this was confirmed histologically, where you get increased hemorrhagic necrosis in the wild type and less so in the knockout. And Lance Pohl's group, which in NHLBI, who used to look at liver toxicity, did histological scores to confirm that the knockout mice in the fasted state had less injury. So we used 2D gel mass spectroscopy to identify potential target proteins. And here you see, comparing the wild type to the knockout, and after mass spec identification, that a mitochondrial aldehyde dehydrogenase, ALDH2, is highly acetylated in the knockout mice and not in the wild type. And ALDH2 catalyzes the oxidation of aldehydes, most commonly known for ethanol, but also for lipid peroxidation products like 4-hydroxy-2-nonenol, which can be oxidized and broken down. And interestingly, also, in a far distant time in 1996, it was demonstrated that after APAP administration, the activity of aldehyde dehydrogenase was markedly reduced by 4 hours, and that correlated with the highest peak in NAPQI concentrations. So the highest levels of the metabolite, the toxic metabolite of acetaminophen, correlated to the lowest expression of ALDH2. So this made us think this is an interesting target to look at. And we essentially, and I guess I'll show some of these data, but showed that ALDH2 is a substrate for SIRT3 deacetylation. This is immunoprecipitation with acetylysine, and showing the ALDH2 levels are increased in the knockout animal. And I guess here I'll show that if you add NAD, which activates SIRT3, you deacetylate the ALDH2, and if you add nicotinamide, you reverse that. And if we use wild-type SIRT3, you deacetylate ALDH2, again using immunoprecipitation assays, but if you use SIRT5 or a deacetylase mutant SIRT3, you don't get this deacetylation. So it seemed that ALDH2 was a legitimate substrate of SIRT3. And then we showed that in the knockout animal, the activity of ALDH2 was retained to a greater degree in the knockout animal. No change in ALDH2 levels, but if you looked at hydroxynonenal levels, again you see with 1 hour of acetaminophen, marked accumulation in the wild-type mouse, and this was diminished in the knockout mouse. So we then used a viral shRNA approach in the livers and demonstrated that when you knock down ALDH2, you reversed the beneficial effect in the SIRT3 knockout animal. And if you look at the histology again, you get increased hemorrhagic necrosis in the knockout mouse when ALDH2 is knocked down versus the scramble control. So we then looked at NAPQI binding to ALDH2 and demonstrated that, in fact, when you ALDH2 does bind to, NAPQI does bind to APAP to ALDH2. This was an IP experiment with overexpressed flag-tagged ALDH2, when you added NAPQI and you measured for the adduct, it was increased. But if you added nicotinamide, which blocks SIRT3, there was decreased binding. And we also show, again, with these overexpression studies, that if you use wild-type SIRT3 and the flag-tagged overexpression of ALDH2 with IP with the flag and immunoblot with the NAPQI, you see increased binding. And if you use the deacetylase mutant, so you retain acetylation of ALDH2, you get less binding. So suggesting that this acetylation of ALDH2, in fact, could be modulating the binding of this toxic metabolite and therefore play a role in hepatotoxicity. We then use mass spec to identify the sites on the ALDH2 that could be modulated by this acetyl modification. And in fact, you get a 42 Dalton change in spectra when the lysine is acetylated. And we found two sites, the lysine 370 and the lysine 377. And when we mutated those to either a glutamine, which mimics acetylation, maintains the positive charge, or arginine, which mimics deacetylation, you see there was no change on the 370 site of the binding of NAPQI, but when you mimic acetylation, you get less binding, suggesting that, in fact, this is the site where NAPQI binds to the deacetylated LDH2, inhibits its activity, and promotes liver toxicity, and that's shown in the schematic here. So this acetyl here acts as a allosteric inhibitor of the binding of NAPQI. You remove it, NAPQI can bind, inhibit the aldehyde dehydrogenase, and you get increased acetaminophen-induced liver injury. So that was an example of fasting and a disease process that's modified by acetylation with the targeting of a specific protein involved in the metabolism of Tylenol or acetaminophen. I'll now go into looking more broadly at the role of certain biology with mitochondrial quality control. But firstly, I'm just going to introduce this concept of the inflammasome. So the inflammasome is a multi-protein intracellular complex that's either sensed as pathogen, which is usually extracellular bacteria or crystals or cholesterol, or damage-associated molecular patterns, which could be intracellular, which activates caspase 1, cleaves pro-inflammatory cytokines, and drives the cytokine cascade. And there are numerous different inflammasome programs, and a central feature is the NOD-like receptor family of proteins. And the second protein involved is the adapter of apoptosis spec-like protein with a chiron domain. So that's ASC. And here you see the NLRP3 with the ASC domain and caspase. They form this multi-protein complex, which enables the activation of caspase for the cleavage of the pro IL-1 and IL-18 to produce the cytokines that drive that cascade. And the NLRP inflammasome is, I guess, most recognized as a component of sterile inflammation, which occurs with diseases like atherosclerosis, diabetes, asthma, and interestingly, with aging, we get increased sterile inflammation. And it's regulated at numerous levels, both at the transcript level and post-translation. So the transcript level is noted is defined as the priming of the inflammasome, and that can be mimicked in a model system by using lipopolysaccharide, which binds to the toll-like receptor, activates NF-kappa B, and activates NLRP3 proteins, IL-1 beta transcripts to generate the proteins. And then you get activation, which is when this complex comes together as this multi-protein complex to drive the disease. And this can be stimulated by ATP, nitrous ricin, which is a bacterial membrane, fatty acids, and cholesterol. And interestingly, and why we got interested in this, is that one of the disease-associated molecular patterns or DAMPs that can drive the NLRP NLRP3 inflammasome activation is damage to mitochondria. In fact, there was a wonderful review by Martin in 2012, which demonstrated that numerous extracellular stresses could drive potassium into the cell, lose mitochondrial membrane potential, and through some mechanism you could get DNA oxidation and release, and this mitochondrial DNA could drive this inflammasome. At the same time, other people have shown or suggested because of the symbiotic hypothesis of mitochondrial beam bacteria, something that either Wynne or Richard alluded to, their DNA is slightly different to nuclear DNA, and that you have these unmethylated CPG motifs, and mitochondrial RNA and tRNA generate N-formyl peptides. And these peptides and these motifs in their own right act as dams to drive the NLRP3 inflammasome. So this was what made us interested in the inflammasome. And interestingly, I'd previously discussed the fact that sirtuins could be activated by feeding and fasting, and obviously suppressed by overfeeding. An interesting study came out from Mark Madsen at the NIH suggesting that alternate day caloric restriction improves asthma. And another disease, rheumatoid arthritis, has also been shown to have less flares if patients undergo intermittent fasting. And in fact, it's become, I think, a big rave. A lot of people do intermittent fasting as a potential health benefit. So based on these findings, the recognition that mitochondrial integrity or lack of integrity could drive the inflammasome, we wanted to pursue this further. And just in terms of the sirtuins that control mitochondrial integrity, one of the nucleus sirtuin, SIRT1, can improve mitochondrial integrity by driving or activating a transcription factor called PGC1 alpha by deacetylating PGC1 alpha and driving mitochondrial biogenesis. And SIRT3, in low nutrient states such as caloric restriction or fasting, can affect many different pathways to either increase metabolism, but also increase stress defenses. Antioxidant programs that activates MNSOD, promotes ammonia breakdown, reduces mitochondrial permeability transition, potentially decreases protein synthesis, and even is suggested, albeit indirectly, to play a role in promoting mitophagy. So these sirtuins could modulate mitochondrial integrity. And so the hypothesis was, can mitochondrial sirtuins regulate the NLRP3 inflammasome? And is this dependent on the modulation of mitochondrial homeostasis? So the first thing, and this was Javier Trabe in the lab, looked at mice where he fasted them for 48 hours as a caloric restriction mimetic, and looked at primary peritoneal macrophages to look at IL-1 beta release, and demonstrated that there was a robust reduction in IL-1 beta release and a similar release of TNF-alpha, which is not shown here, suggesting that this could be an NLRP3 associated event. He then used human THP1 macrophages, where he knocked out SIRT1, 2, 3, and 5 sequentially, the mitochondrial ones as well as the nuclear encoded sirtuin that can promote mitochondrial biogenesis, and demonstrated that in fact there was the depletion of SIRT3 that could activate IL-1 beta, and again the release of TNF-alpha was similar in all these cells. He then looked at overexpression of SIRT3 and showed he could decrease IL-1 beta release, and when he used the deacetylase mutant, this effect was attenuated. He then repeated his fasting studies comparing the wild type and the knockout animal, and showing that in the fed state there was no difference, pretty similar to in fact what we saw with the liver and acetaminophen. But in the fasted state there was a much more robust reduction in IL-1 beta release in the wild type mouse compared to the SIRT3 knockout mouse. And in fact, this was replicated at the level of IL-1B, the cleaved protein, showing it was higher in the knockout and less so in the wild type. And he replicated this looking at bone marrow-derived macrophages to show that there was higher IL-1 beta with no change in TNF-alpha. The story is a little bit more complex than I'm showing here because, in fact, there's a different effect on feeding and fasting, on priming, and on activation, which is a mitochondrial event. But we reduced the talk somewhat, and that I'm not going to show that data. But nevertheless, with respect to the mitochondrial homeostatic role, we've already discussed all the beneficial effects on the mitochondria. And Javier shows that if you knock out SIRT3 in the THP1 cells, that indeed you get increased mitochondrial reactive oxygen species, and this is replicated by rotenone. And then he looked at mitochondrial release into the cytosol. And when you take wild type, and these are bone marrow-derived macrophages, and you add LPS and as a prime and then ATP, you get a very modest release of mitochondrial DNA into the cytosol. But if you use the SIRT3 knockout mouse, when you add the ATP, which activates, it's the activation component of the inflammasome, you see this huge increase in mitochondrial release into the cytosol, suggesting that the lack of mitochondrial integrity by the disruption of SIRT3 could play a role in this inflammasome process. So a previous postdoc in the lab, Ian Scott, was looking at trying to find proteins that counter-regulated SIRT3 and had identified a component of the mitochondrial acetyltransferase program called GCN5-like1. It's not the complete enzyme system, but it does play a role. And interestingly, as I'd shown you previously from Eric Burden's lab, with fasting, SIRT3 goes up, and GCN5, and this is work from Brad Webster, is profoundly reduced in the fasted state compared to the fed state. So they're reciprocally regulated in this nutrient specific pattern. And in fact, they do show counter-regulatory effects. There, if you knock out SIRT3, you get increased mitochondrial protein acetylation, and this is lost when you knock out both proteins together. And Brad Webster had shown that if you knocked out GCN5-like1, which actually could be associated with a slight increase in SIRT3, you seem to have increased accumulation of LC32 and P62 in the mitochondria, which could be associated with increased mitochondrial mitophagy, and he went on to show that the mitochondrial number's down. But if you knock out SIRT3, you lose this effect. And in fact, with the knockdown of SIRT5, you also get increased ubiquitination of mitochondrial proteins, and effects seem to be reversed by the knockdown of SIRT3. So suggesting these proteins have counter-regulatory effects. So Javier looked to see what happens in the GCN5 knockdown macrophages, and demonstrates that as opposed to SIRT3, you get a profound reduction in IL-1 beta, although interestingly, you also get some reduction in TNF-alpha, which is not an inflammasome-associated event. 5. It's so GCN5 itself is a nuclear acetyltransferase, and GCN5-like1 is mitochondrial enriched, and it seems to be a component of a mitochondrial acetyltransferase machinery, although in isolation, or if you overexpress it alone, you can't change acetylation. So it's probably a regulatory factor in that program. Sorry, I just used it as a counter-regulatory and didn't explain it in detail. And again, if you looked at reactive oxygen species, it's down in the GCN5 knockdown, and by adding calcium to the cell as a means, incrementally adding calcium to look at a resistance to permeability transition, the GCN5 knockdown are much more robust, again suggesting improved mitochondrial integrity. So as we got into this field because of a human study showing that fasting could, intermittent fasting could attenuate asthma and flares in rheumatoid arthritis, we wanted to see if this inflammasome was indeed regulated in human subjects. And we're still looking for a few volunteers to finish this one off. And we do have we have an asthma protocol at the IRB at the moment to actually study the disease. But this was in normal controls. And we had the individuals eat a 500-calorie meal. They came to the clinical center. We sent them on their way for 24 hours, and all they could do was drink water. And we did blood tests. We did glucose and insulin and fatty acids and growth hormone and ketones to check that they were fasting or not. We did a fasting blood draw. They had another fixed caloric meal, and we reassessed we drew blood again at 1 and 3 hours so that we can compare inflammasome activation and mitochondrial function at these 3 time points. And I guess the time's much better than I thought. But anyway, so these are the data, at least on IL-1 beta release. And you see that even without the addition of LPS, so purely in the basal state, when you stimulate the inflammasome, the fasted human subjects, both peripheral blood mononuclear cells and monocytes, have markedly less IL-1 beta release compared to the 3-hour fed state. And if you look at trying to prime the cells with either 0.1 or 1 of LPS, you see, albeit much less so, higher IL-1 beta release in the 3-hour fed state compared to the fasted state. And this is associated with increased mitochondrial reactive oxygen species, less oxidative capacity, suggestive of a change in mitochondrial function. And we've now added data using a SIR3 agonist, suggesting that we can reverse this effect. But I'm not showing that data today. So in conclusion, the NL of pre-inflammasome is blunted by fasting. It's is activated in a nutrient level dependent manner, and it's modulated in part by the SIR3 GCN5-like-1 regulatory program. And preliminary data suggests that fasting and mitochondrial acetylation regulatory programs modify the role of mitochondria as a damp in this activity, and it's operational in healthy human subjects. And as I said, we're going to look at asthmatics next. So just a final schematic. SIR3, I guess we'd have to call it Jekyll and Hyde, because my first story suggested that acetylation was beneficial, and that's where the acetyl moiety on the lysine could sort of fend off, in our case, a reactive metabolite, but it's also been shown to fend off ubiquitin or chaperones involved in chaperone-mediated proteasome degradation. And the flip side is that under fasting or nutrient sensing conditions, you can get an increase in SIRT with a decrease in GCN5, deacetylation of mitochondrial proteins, improved homeostasis, and protection from diseases where mitochondrial integrity can exacerbate it, such as the NLRP3 inflammasome. And so to end off, I just wanted to acknowledge the people that did the work. Zhongping Liu did the acetaminophen story, and he's now started his own lab at GW. Ian Scott was the GCN5. Brad Webster also worked on GCN5 and Autophagy. And the preliminary data today is predominantly done by Javier Traube with help from Jessica, Miriam, and Kim. Thank you very much.
Thank you very much, Michael. Well, yes. Okay, sure.
Thank you. Question is to the first speaker. So you mentioned that certain
Richard, why don't
diseases
you...
occur, blindness, in this retinitis pigmentosa, or some other disease?
He wanted to know if it was retinal, what?
Method about
The blindness is called DOA, dominant optic atrophy. It's a loss of retinal ganglion cells. But I had a question for Michael. So it's pretty interesting that the inflammasome NLRP3 is inducing the release of mitochondrial DNA.
Yes. Well, no. So it's the release of the DNA activates, is thought to be a damage-associated pattern, which then activates the coalescing of all these proteins to generate the inflammasome.
And so you don't see it unless you add ATP.
ATP,
I thought
right.
the ATP was activating it to cause the release.
So we've used,
what were you measuring? What was on the y-axis?
Right, so there was IL-1 beta release.
And you have to add ATP to get IL-1 beta? Is this in a cell-free system?
That's in a cell, well, it's in a cell culture system. We can also use nigericin
You add ATP to the media
to the media
to activate the inflammasome?
to activate the inflammasome.
Isn't there plenty of ATP already in the cell to activate the inflammasome?
It seems to, I guess it promotes a huge influx of potassium. But we also use nigericin, which is another trigger, and show the same.
Oh, I see. So
right.
it's not really about the ATP.
ATP is also a dam.
Wait a
Yeah,
minute, wait a minute.
right.
Gotta,
Right. So ATP,
you
yeah,
wanna make
itself
a
would also be a
comment?
dam.
no, but they couldn't hear you, so
ATP is also a DAMP.
you. There are other questions? Yes.
Has anybody actually measured aldehyde and acetaldehyde or other aldehydes after Tylenol?
So the,
If you inhibit the dehydrogenase, you might expect an increase in the level of aldehydes, right?
I'd imagine it has been done. I don't know. I do know that there are now small molecule activators of the aldehyde dehydrogenases, and they also have these protective effects. And the enzyme itself has been shown for many years to be suppressed by higher doses of Tylenol.
Another question. How do you exclude acetylation of all the other proteins when you're looking at your acetylation?
Right. So we don't. We don't. We demonstrate that this one's... well, first of all, aldehyde dehydrogenase is an important player in the breakdown of the reactive metabolites of acetaminophen, because we show when we knock down the ALDH2, we lose the protective effect. But it is a very reductionist approach. I'm sure there's 5, 10, 40 other proteins that could also be targets for either NAPQI or other toxic intermediates that this could have an effect on. So I don't think it's the exclusive, but it is important in that process.
So,
Well, the IL-1 release, a very small amount of increase of the IL-1.
The problem is that some of the DAMPs are so powerful that, and so nigericin is actually a better way to look at the data, but because that's why you lose the effect of priming, because high ATP is very damaging to the cell.
so you think that the Tylenol package should also say, don't eat on an empty stomach?
But there are a lot of case reports of people doing religious fasting, many different religions, who then take Tylenol and get hepatotoxicity. And as I said, that big study out of Pittsburgh suggested that you needed a lower dose, but the mechanism hadn't been figured out. And I guess it's just, we're a society of overabundance. So I think it's actually usually patients with appendicitis or reasons why they're not eating, where then they take that Tylenol and they get into trouble.
I think most people on severe caloric restriction voluntarily, they all have headaches, and they probably pop a few Tylenols and it accumulates, which is
It's pretty
possible. Yes.
So there are people who argue that if you vaccinate in the morning or if you vaccinate at night,
Right.
you'll get different responses.
Right.
Do you think that amount
Right,
of fasting just overnight?
Right. So we've discussed that, and I spoke to Richard Siegel about it. Different, Richard. But so the inflammasome plays a role in the alum in the vaccine activates the inflammasome, and therefore you can change your vaccine response. So the question is, should we, after the asthma study, should we be doing a study in octogenarians who have a very poor response, and see if feeding or fasting will change there?
What about just the fact that you don't eat for 12 hours? Just
fact that you don't eat for 12 hours, and in the morning you will be more
Right.
fasted than you are at night.
Right.
I mean, you
So-
did 48 hours, but how long does it take?
No, well, that was the people were 24. We couldn't.
24, okay, but would 12 do?
The ILB wouldn't give us 48. But no. But so the so I would speculate that you might have a better response. No, on a fed, right.
Right, at night, after eating breakfast, lunch, and dinner.
Well, right. So there's also, this whole thing about what happens to, acetylation. If you look at it, there's the clock plays a role, and you get a change in. So that would be adding another dimension, which we haven't looked at either. But I would speculate that you could augment the response.
So I know one bacteria that lives inside my
Talk into the
macrophage, TB. So cellular system has a stored incorporated bacteria, which is the mitochondria. So is there anything we could learn from the TB bacteria in my macrophage? What happens to mitochondria? How much changes occur?
wait a minute.
So Jeffrey Cox had a paper in Nature 16 months ago saying Parkin translocates to typhimurium. Is that it? It's a mycobacterium, typhimurium mycobacterium. But he said, he's sort of like, you can't really test it because it's got some kind of type 3 secretion system. And most of the strains we could all use in a normal laboratory are blunted in some respect. And he's got like a P3 facility at UCSF, and he's got the full-blown TB, and it translocates there and not in the others. So I think the world's watching whether there's a link between mitophagy and it's called xenophagy, where you have autophagy. It's definitely a host defense pathway against bacteria, where you use the autophagosome. And what's the recruitment? Clearly, ubiquitination of those typhimuriums are involved. And we're now screening. I've got a Shireen Saraf in my lab is now screening for E3 ligase involved in those pathways. There's clearly E3 ligase. There's a Parkin, I don't know.
Wait a minute. People are attending, that's why.
Sorry, we can move up.
No, it's not them.
It's
It's 200 people
I
who are
see.
listening, and they want to hear you.
You've got an intracellular bacterium. Autophagy is a great way of getting nutrients for that bacterium. So is that part of the defense mechanism, or is that the bacterium telling the cell to do this, to eat itself, to give the bacterium more nutrients?
Yeah, that's, there's so many different pathogens. There's probably 50 different ways. And it's a cat and mouse game. Whatever they're doing, we're counteracting that, or we wouldn't be here. So it's sort of at a stasis. But I know viruses are also very complicatedly involved in these pathways. And some of them actually stimulate autophagy, and they bud off of the autophagosomal membrane. They're using it. But maybe sometimes we use the autophagosomes against them. It's probably all of the above.
Parkin a nuclear gene?
Yes. Almost everything is a nuclear gene. It's 33,000 to 13. All the mitochondrial genes are either tRNAs or they're in the OXPHOS complex. They're embedded in the inner membrane.
All right. Well, listen, thank you very much. This has been quite an eye-opener on many fronts. We appreciate it.